So the next one is physical properties of amino acids. The section says physical properties of amino acids. The first one, whether they are white in color, blue in color, how are they? Amino acids are colorless. They are non-volatile crystalline solids. Okay, if you have some substance and if I ask you this substance is soluble or insoluble in water, how to guess? So if any compound is soluble in water, if it forms some kind of charge over it, or if it is ionic in nature. So for example, NaCl, all the ionic compounds like NaCl exists as Na plus and Cl minus. So NaCl is soluble in water. In the case of amino acids, you have NH2 and COOH group. In water, it might happen that this NH2 will gain one H plus ion to form a NH3 plus, and this may lose a H plus ion to form a COO minus. So it is ionic in nature. So anything which is ionic in nature is soluble in water. So soluble in water. The NH2 group, which is a basic group, and COOH group, which is an acidic group, neutralize each other. Acid plus base is the reaction of neutralization. So what happens is that this RCHNH2 COOH actually exists as a neutral compound like this, NH3 positive and COO minus. So you have one anion and one cation. Fourth point, the isoelectric points of alpha amino acids. Isoelectric points of alpha amino acids. So suppose your amino acid is this. The case one is, suppose you put this alpha amino acid in acidic medium. So the medium is acidic. When is a medium acidic? Any medium which contains more of H plus ions, it is acidic in nature. Will H plus come and react with COOH? Will acid react with one more acid? No. Will acid react with a base? Yes, it will. So in the acidic medium, it forms a compound, forms RCHNH3 positive and COOH. Now as a whole, this compound has a charge of plus. So this forms a cation, and suppose if you apply an electric field, this ion will tend to move towards the cathode electrode. Suppose in a similar way, I put this amino acid in a basic medium. In a basic medium, you have more of OH minus ions, or you have a deficiency of H plus ions. So if you have a deficiency of H plus ions, the basic medium will not react with one more base. It will react with an acid group, and it will form CHNH2 COO minus. Now this group is an anion, it has a negative charge, and if you apply some electric field, it will go towards the anode electrode. Anode is the positive electrode, cathode is the negative electrode. Opposite charges attract to each other. Simple. Now, the acidic medium has a pH of 1 to 5. The basic medium has a pH of 8 to 14. Suppose in between the two, if I have some pH level at which what happens is that the two effects, that the positive charge and the negative charge, balance each other, then the overall compound will be neutral in charge. So in that case, we use a term called as isoelectric point. I'll just erase everything. So it is a pH at which there is no net charge over the amino acid, and this pH level is known as isoelectric point. So if there is no charge over the amino acid, it will not migrate towards either anode or cathode. The fifth point, whenever we analyze a compound, we always see whether the compound is optically active or not. I must say one thing, that optical activity should not be in physical properties. It is a chemical property. But let's learn. Suppose I write the like this, hydrogen NH2. When is a compound optically active, or when is a carbon known to be a chiral atom? Whenever a carbon is attached with 4 completely different groups, the overall compound is asymmetric and the overall compound comes out to be optically active. And it is in the case of alpha amino acid. So we see that alpha amino acids are optically active in nature, and the alpha carbon is okay. So we can name these amino acids as either D or L compounds. We see one thing, that all the amino acid, the major amino acid, the top popular ones, they all are L compounds. So we sometimes also refer to them as L alpha amino acids. So that's all about the amino acids. In the next class, we will learn about the when we add amino acids, we form peptides. So we will learn about them. What are these?
Thank you for watching. Visit us at needbook.in.